My name is Fini, and I am presenting a longitudinal comparison of microeukaryote diversity in contrasting urban reservoirs. I took samples from two urban reservoirs on Hofstra University's campus, which is located on Long Island, New York. One of them, Site TP, short for turtle pond, had turtles and plenty of fish in it. Meanwhile, Site SG did not have as many fish and was not home to many other visible organisms. Meanwhile, Site TP was surrounded by a lot more foliage than SG, and SG was encased by concrete. Moreover, Site SG had much more anthropogenic interference, and Site G was in much more shade than TP. In other words, TP had much higher sunlight exposure, and for this reason, I predicted that there would be more autotrophic organisms in Site TP, and there would therefore be higher chlorophyll concentrations. I also predicted that Site TP would have higher microbial diversity as a result of its different environment compared to SG. First, I filtered and extracted my DNA by passing my water through a filter and then suspended it in a buffer until amplification and sequencing. Then I used PCR amplification on the 18S gene to make it much easier to sequence. And then I sent these samples to University of Connecticut in order to fully sequence the DNA and to figure out which organisms are present in my samples. Then, in order to fully evaluate how many autotrophs were in my sample, I measured chlorophyll A by putting it in 90% acetone, and then I measured the concentration of the chlorophyll by seeing the color of it, and the more green it was, the higher the chlorophyll concentration, and then I used these calculations to estimate the chlorophyll concentration in each sample. Then I fixed my samples with 2% Lugol and 1% formaldehyde in order to stain cells and improve visibility under the microscope. And then I utilized both inverted and fluorescence microscopes to categorize organisms and to count them. Here are my microscopy results. As you can see, the two most common types of algae that I found were Sphaeropileus algae and Desmodonis algae. On the top, you can see that's my inverted microscopy results, and that is just regular microscopy. It allows you to see where these organisms are in the samples. However, it doesn't allow you to see unique features such as DNA concentration and chlorophyll concentration. And so on the bottom, I use fluorescence microscopes, and the red-orange that you see, that is the chlorophyll, and the blue that you see, that is DNA. So as you can see the nuclei in all the eukaryote organisms, and you can also see the autotrophic organisms because they are red as a result of chlorophyll. And so the main organisms that I found were the green algae, the diatom chains. I found a lot of bacteria, especially near organisms such as green algae, and I also found plenty of amoebas. In Site SG, green algae was dominant in both 2024 and 2025, and the same trend was found with diatoms and TP in both years. However, both sites show significant signs of stress. As you can see, dinoflagellates, which are very sensitive, seemingly disappeared between 2024 and 2025. The same trend was found in TP. As you can see, a lot of the diatoms were lysed, which once again is an indicator that these ecosystems are being stressed. The Shannon Diversity Index is a very good tool to help categorize different ecosystems and seeing which ones are more diverse than others. And one thing that was really shocking in my experiment is that SG, which had much less sunlight and more anthropogenic interference, actually had a higher diversity index score than TP. In fact, this difference was significant with a p-value of less than 0.05. Figure 10 is a great way to help visualize these results. As you can see, the big green bar represents green algae, and TP, the green algae between both years, continues to dominate the ecosystem. However, in SG, while this green algae did play a role, the diatoms played a much more expansive role. As you can see in 2024, that yellow bar, which represents diatoms, had a very significant portion of the biomass. As you can see, compared to the estuaries, these urban reservoirs are very unique compared to other ecosystems. So for my discussion, algae dominated both sites, obviously, but there were some differences. SG was rich in green algae, meanwhile TP was dominated by diatoms. Both sides showed many signs of environmental stress. For example, TP had increased lysed diatom material. Also, SG had a complete disappearance of sensitive dinoflagellates. SG also had way higher diversity than TP, which was not expected. Water quality and habitat structure were stronger predictors in diversity than shoreline type alone. And also, reservoir communities were distinct in estuarine controls, showing more freshwater adaptation. For limitations of this project, only one sampling event occurred per year, and this prevented me from fully analyzing seasonal succession. Also, future research should vary the depth of the samples, as surface-only sampling might alter the results. Also, metabarcoding detects DNA from both living and dead cells, which could alter my results. So in conclusion, while recent research has neglected urban reservoirs, they cannot be overlooked because just the visual characteristics can severely misrepresent what happens on a microbial level. And future studies should help address the limitations of my project. For example, a big thing is capturing seasonal succession. This is my works cited. Thank you for listening.